Hello ladies and gentlemen, and welcome to Mr. Saucedo's YouTube videos. Today we're going to be going over indicators, pH probes, buffers, and titrations. So make sure that you're following along, and if you have any questions, obviously make sure that you ask. So what are we going to talk about first? We have to talk about pH probes because the easiest way, without a doubt, to measure the pH of a solution is to use a pH probe. And here's the thing, our pH probes, they look kind of cheesy and cheap, but they're really not that cheap. In fact, this is my sort of crappy drawing of what a pH probe looks like. It is a red plastic pH measuring device. There we go, that's a good way of saying it. It has this weird black sort of tube thing, and then it has if you look inside, a glass electrode which converts, sort of electrical potential stuff into a pH reading for us. And it is without a doubt, sort of the easiest way to measure the pH of a substance, but sometimes they are not available. And so in that situation, when they're not available, scientists resort to indicators, which we've been using for literally thousands of years. So what are indicators? They are chemicals that change color in the presence of either H3O+, if we're talking about acidic stuff, or OH- if we're talking about basic stuff. So here's the thing about indicators. You've seen them before. At the very beginning of this unit, I showed you examples of some indicators and the color changes that they undergo and all of that, and I said we'd talk about it eventually. Well, we're at the end of the unit, so we might as well talk about them. What you see here on this weird chart thing, these are all different indicators, and yes, they have like horrifyingly complex names, at least some of them do, but they come in a variety of colors and they work in a variety of pH ranges. So what you see here is a collection of some of the most widely used indicators for scientific purposes and testing. So we've got, things like methyl purple, which have a very slim sort of pH range and a very specific pH at which they change. We have things that are very general, which I just kind of covered up, but like litmus paper and stuff along those lines. We've got phenolphthalein, we've got methyl orange. I mean, there are a lot of really cool indicators out there, and we're going to be using, just a handful of them. So what do we need to know? Well, if we're going to pick an indicator to see what the pH of a solution is, we want to make sure that we choose an indicator that changes color in the range that we want it to turn in. So if we know that, the pH is going to change, I don't know, around a pH of 10 or something, I would not want to pick any of these guys because they don't change color around 10. But this guy right here seems to be one that would probably work, okay? And so that's the idea. So if you are smart with the indicator that you choose, then you can actually get the indicator to do most of the work for you. So that's kind of nice. Again, if you wanted something a little bit more acidic, let's say we wanted something with a pH of 6, like we know the pH is going to change around there. So litmus paper is a good choice. We've got this, we've got this. We've got a whole bunch of different choices at different pHs, so that's kind of the point. Now, what's the one that we're going to be really using? It's this guy right here. And so he is found right there. Phenolphthalein. Thankfully, you don't have to know really how to spell it, but phenolphthalein, it is probably the most widely used, it looks like I crossed it out, it is probably one of the most widely used indicators in all of chemistry, and it changes bright pink in base, and it changes clear in acid. So that's a very important fact to kind of remember. So I know this seems like a little tangential thing, but we have to kind of talk about buffers. Buffers are important in biology, and so this is really the only time it's actually appropriate to talk about them. So what is this weird buffer thing? It's a mixture of a weak acid and its conjugate base. So for example, a weak acid like, vinegar, HC2H3O2, its conjugate base is C2H3O2-. So if we had a mixture of these two things, of just, sort of pure vinegar and acetate ions floating around in it, we would have effectively a buffer solution, and that works with any weak acid and its conjugate base. Now what makes buffers so special is that they drastically resist a change in pH. So even though, pH sort of varies and changes and everything, if you have a mixture that is a buffer, if you add a little bit too much acid, so let's say I'm adding acid right now. If I add acid, the conjugate base will react with it, and so it'll kind of neutralize things out a bit. If, on the other hand, I add base to this, then it will react with the acid and it will neutralize it, and we'll still have kind of a neutrally based solution. And so the reason why they work is because it contains both a possible acid and a possible base that can react with one another. And so it makes it kind of a useful way of being able to monitor and control things inside of a solution. And in case you haven't figured this out yet, that's exactly what our blood does. Our blood contains a buffer system which helps us survive because our body survives in a very narrow pH range. If our blood gets too acidic or gets too basic, our bodies will cease to function and we could actually die. So we actually survive in a very narrow pH range when it comes to our blood. So what does our blood use? It uses a buffer of carbonic acid and hydrogen carbonate. And so in that space, which I just spent a long time erasing, we're going to write down sort of like a brief little buffer reaction for what these things look like. So let's start with what would happen if you added a base to something. All right, so we have our acid here, which is carbonic acid. And let's say we are adding now a base or something with OH minus in it, okay? Then what I would end up with, and I'm going to use a double arrow for this, is H2O forming because I would have my OH and one of my H's sort of coming from my carbonic acid, and I would make bicarbonate like this. And by the way, that little one, that's a 3. It doesn't look like a 3 though. That still doesn't look like a 3, but that's about as good as I'm probably going to be able to get it. So this is an example of kind of how a buffer solution would work. So my H would react with the OH minus, and I would end up making some water and making some bicarbonate. Why is that important? Because I'm making bicarbonate, and bicarbonate is another part of my buffer. So I'm basically just making my buffer, a little bit different than it was to start with. Next up, though, let's say that I have my bicarbonate like this, and now I'm going to be adding acid. Well, in the process of adding something like an H+, okay, I get H2CO3. So again, by my acid reacting with my conjugate base, I end up making more of my carbonic acid. So they make each other in their reactions, which is why they're so good at resisting changes in pH, because not only does it react very well with acids and bases, but whenever it does that, it makes the opposite component. So it makes more conjugate base, or it makes more weak acid. And so it actually ends up being a pretty useful device, especially inside of living things. The last bit here gets a little complicated, but it's only complicated because we haven't done a titration yet, and we'll get to do that very soon. So here's the thing about titrations that you need to remember. A titration is just a method, okay? It is just a lab technique for determining the concentration of a solution by reacting a known volume of that solution with a solution of known concentration. So in other words, we are going to be reacting something that we know the concentration of with something we don't know the concentration of, okay? And by figuring out how much it takes before the reaction occurs, we can determine the concentration of our unknown. And the way we kind of graph this is we look at pH over the volume of acid or base that has been added. And the weird thing is you always see this same curve. Not necessarily in the same spot, obviously, but you see the same general shape. These right here are what we call titration curves. And it's something that is probably, again, ubiquitous with acid-base chemistry. It's something that always shows up. So what are the major features of a titration curve? Well, the first key feature is called the equivalence point, and it's the point at which the moles of H plus and OH minus are equal. And so on a titration curve, so again, I'm going to just sort of shade this in, it occurs directly in the middle of this gigantic change in pH. That's our sign that our reaction is done, is when we see this gigantic change in pH. Notice that actually right here at the bottom of each one of these curves, it's actually a buffer. So it's not really changing in pH very much, and then all of a sudden, whoa, it just increases dramatically, and then it starts to buffer out again. And it's the same thing. So this is a buffer, and then all of a sudden, whoa, it increases like crazy because our reaction occurred, and then it starts to flatline at the top again. That right in the middle here of each one of these curves is called our equivalence point. Now notice, again, you want to pick an indicator that changes color in the range where your equivalence point is. So if you know your equivalence point is going to be, somewhere around, 7, bromothymol blue is a really good choice because somewhere along this line it will change color. Same thing goes with this over here. If our equivalence point is going to be like around 9, phenolphthalein is a great choice because somewhere along this line it will change color. And so the other key point actually isn't really a point on the graph. We call it the end point though, and it actually is the point at which the color changes in our reaction. So technically, as I erase kind of my writing here, that weird little color thing that you kind of see, this sort of box here, of color, this is where our end point is. It's going to change color somewhere in that box, and depending on how good your eyesight is, it might be different for each person. So some people will notice the end point really early in a reaction. Other people, it'll take a little bit of time. So those are just the key areas, though, on our titration curve. If you have any questions, make sure you ask.